Okay, well, welcome everybody. Thank you for joining us. Today we have the Andover High School BioBuilder Club team here to share about how they have used BioBits in their projects in the last couple of years. And I'm very glad that they are willing to present this because I think BioBits is a tool that could be really helpful to a lot of schools based on your needs in the high school classroom, being able to conduct this kind of laboratory research. And their use of BioBits has been very successful. So I will hand it over to them to tell you about BioBits and their project. And then if anyone has any questions, I am sure that they are happy to help. So
Yeah,
take it away,
and I think
guys.
feel free to chime in because I don't know if I'll be able to see the chat while
Oh,
we're
okay.
sharing. So just jump right in.
Perfect.
Great.
Um, so just for like a little quick introduction, I'm Ella.
I'm Tommy.
I'm Siri. And I'm Kirsty. What next? So just a brief overview on what we're going to be talking about today is a little bit of background, what BioBits is, our design and expression of DNA, and then
It's a
some
little
time.
bit hard to hear you guys. I'm not sure where the microphone is. Sorry to interrupt.
Loudest you've ever spoken.
So this is just going to be a brief overview of our project. We're going to discuss like what is BioBits, how does it work, our design and expression of DNA using BioBits, and some challenges, limitations, considerations for our project, and then at the end, our results. So for a quick overview, some facts you might want to know is that approximately 400 million tons of plastic is produced annually, and polyethylene terephthalate, or PET, makes up about 14% of that. But only 5% of PET is actually recycled. 15% is incinerated, which releases a lot of CO2 into the atmosphere, and 75% ends up in landfills. So we use an enzyme, PETase, which breaks PET down into MET, or monoethylene terephthalate. From there, another enzyme, metase, can break the MET down into more environmentally friendly compounds. So with these two enzymes, PET can be broken down into more eco-friendly substances. There's another enzyme that we use, leaf compost cutinase. We use two forms, which can depolymerize PET in one step. Unlike PETase, it's more thermostable, and which is great because it can withstand the high temperatures of recycling facilities, which can typically range from 55 to 60 degrees Celsius. And in our project, we used engineered forms of LCC. There is an old one that's not very thermostable, and a new one which is much more thermostable. And PETase to enhance the biodegradation of PET. And then using BioBits, we expressed the enzymes and then added two forms of PET, film and powder, to the resulting protein. So, what are BioBits? BioBits are freeze-dried biological compounds. So instead of using living cells, scientists take the working part out of cells and freeze dry it, which is what BioBits are. This process stops all biological activities, but it still keeps the biological parts of the cells intact. These dried components can be stored for long periods of time and activated again by adding water. So this will allow them to carry out transcription and translation without living cells. And when this is done properly, BioBits will produce color, which signals protein synthesis. So what actually takes place in BioBits? So once water is added, it reactivates the freeze-dried system in BioBits. So first, DNA is transcribed, which makes messenger RNA, and once mRNA is translated, it makes protein. So if it's a fluorescent protein, the protein will glow when light hits it, which signals that proteins are being created. Compared to traditional methods, which requires living cells and requires you to perform transformation and other steps, using BioBits as an alternative is way less time consuming and much easier for our high school project. And so for us, using BioBits worked on our first experiment when we expressed our enzymes. So how are BioBits a part of synthetic biology, because you are designing and building and testing a biological function.
So how did we use BioBits? We use BioBits to express our enzymes. As Ella mentioned, DNA is transcribed and translated using BioBits. And then we added our DNA at multiple concentrations to test for fluorescence. According to the BioBits co-creator, reporter or colored proteins do well at 1 to 5 nanograms per microliter, and enzymes do well at 25 to 50 nanograms per microliter. So we monitored our BioBits for over 9 hours, and we took pictures at regular intervals. We then used these pictures to quantify the fluorescence differences using ImageJ. We did have some inconsistencies between our controls due to pipetting errors. So we had our plasmid designed and then ordered from Twist. We had his tags added if we wanted to purify the proteins, but this is not something that we have tried yet. T7 promoters and terminators are the viral parts that encourage BioBits to do transcription. It is the specific start signal that the T7 RNA polymerase in the BioBits looks for. However, this is also a limitation because if we were to move this plasmid to a cell, these parts would be so strong that they would cause an unequal distribution of resources in the cell.
So we first designed the plasmids and then sent them Twist. And Twist had to grow the DNA inside live E. coli bacteria, but initially they were unable to deliver the PETase plasmid. We also had to perform something called codon optimization, in which multiple DNA codes or codons can code for the same amino acid, and E. coli prefers certain codons over others, and our enzyme PETase comes from a different bacteria. So the codon optimization rewrites the DNA sequence using the codons that E. coli can accept without changing the final protein structure.
So we expressed our plasmids in BioBits for 24 hours, and then we added PET plastic in the form of powder and film. So the GFPs did not glow at 100 nanograms per microliter, but they did glow at lower concentrations. So what we did was we took 7 microliters at 100 nanograms per microliter, and we let them sit in the BioBits for 24 hours at room temperature. And then after those 24 hours, we combined 3 reactions for a total volume of 21 microliters, and then added our PET film and powder into those 21 microliters. And then those sat for 5 days, either at room temperature for the PETase or at 72 degrees Celsius for the LCC. And then we later rinsed and dried them in preparation for the scanning electron microscope or SEM. So the reactions that were kept at 72 degrees Celsius are the ones that had LCC in them. They precipitated. So you can see in the photos at the bottom, the precipitate that formed and then later settled at the bottom. We separated some of this gunk to observe that under the SEM. And after doing so, we think that this was BioBits that got reactivated. So this is an image from the SEM. So we think that these are BioBits. The bubbles coming out are due to the SEM vacuum.
So we also want to discuss the limitations of using a cell-free system. So first, regarding our GFP tag. So the GFP we attach to the very end of the enzyme, and this confirms that the ribosome successfully read the DNA and produced a protein. But it glowing up does not guarantee enzyme activity, and it's possible that the GFP portion folded correctly and it glows, but the enzyme protein misfolded because of the bulkiness of the tag. And we're also adding plastic directly into the BioBits, like soup, you can call it. So it contains all the other bacterial proteins and salts. So during incubation, these things can stick to the surface of the plastic. And so, for example, when we're using the SEM, it could be difficult to distinguish between the actual enzyme degradation and dried BioBits residue stuck to the surface. Also, we chose not to purify the enzyme which means we do not know the exact concentration of the enzyme. As Siri mentioned earlier, we did attach a his tag, but we're not using it currently. That's kind of complicated, but it's something we're considering for the future. But right now we can't quantify how much enzyme is in the tube and calculate the specific reaction rate.
And here are our results. So we were able to partner with UMass Lowell and their core research facilities to visualize our results using a scanning electron microscope. So this is for the film and the buffer, and as you can see, the scans for the three enzymes are visibly different from the negative control. You can see these little like abrasions or wrinkles. They almost look like they're kind of melting. And this is for our plastic film and water. Kind of the same thing as before. You can see these lesions that aren't present for the negative control with the enzymes. And then those ones that Kirti just showed are from this school year's experiments. This slide has our scans from last school year's experiment, and we were able to use ImageJ, which is a software, to help analyze to see whether the enzymes did degrade the plastic particles. So if you go on the next slide, we have the table, which we calculated or we analyzed through ImageJ, with 1 being the most circular and 0 being the least circular. So with these numbers that we got, we took the difference from a negative control, which allowed us to see how much the enzymes help degrade the plastic.
So to sum up, so our genetic circuit was built on the PGL1 vector, and we used the strong T7 promoter and terminator to match the T7 RNA polymerase present in the BioBuilder system. We also got some interesting results regarding the DNA concentration. We found that at lower concentrations, we observed clear GFP fluorescence, but at higher ones, the fluorescence disappeared. But despite the lack of fluorescence at these high concentrations, our SEM images still showed plastic degradation, which suggests that the enzyme was still being produced and was active, but maybe the JFP tag misfolded at high concentrations, which is why it lost its ability to glow. We also acknowledge the fact that this is a crude system. We didn't purify the enzyme. We did not isolate the protein from the BioBits mix. So we can't really calculate specific activity and efficiency of the enzyme.
If you're interested in knowing more or reading more about our project, this is a QR code to biotech, which we have our paper. And yeah.
And these are our citations. And thank you to our mentors, Dr. Lakayer, past HS BioBuilders, because this is like an ongoing project, and all the other people that helped us. And if you have any questions, feel free to email us. Try and respond.
Do you have any questions?
There's a question in the chat. You want to unmute yourself and ask the question?
Hello. I'm so sorry. I'm on a bus right now. Can you guys still hear me?
Yes.
Okay. I'm really sorry if this is a stupid question. I don't know if I'm not really good at biology, but I am interested in it. So I just wanted to ask, how does the higher enzyme folding cause decreased glow? I know it does, but like, how does that work?
I think we don't know. It must be something. We're just speculating. Something at higher concentrations that's inhibiting the expression.
Oh, of the glow?
Yep.
Is it like one of the genes? Like, is it like stopping from like it was like an unintended consequence?
Yeah,
I'm so
because
sorry.
you can see on this. I don't know if you can see the slide here. So this is.
I can see.
It goes from 50 to 25 to 10 to 5. And you can almost see there the decreasing glow. So you would think if you added another one at 100 that there would be more, but there was no glowing. So we don't know.
Could it be possible that
now,
I put it in the chat, but I know you can't hear or can't read it, sorry. Could it be possible that the fact that it's so tightly folded could prevent transcription translation from happening and prevent that expression?
It is possible, yeah. Good question.
Definitely possible.
I have a question for the team. So I know that kind of your use of BioBits is from my understanding of BioBits kind of novel in terms of the what you're trying to do and the results that you got, because a lot of the high school projects aren't necessarily set up like this to get results from the SEM. So I guess my question is like is there information, because I'm not super familiar with BioBits. Is there information from other groups that have used BioBits and kind of like how their results varied?
Like you said, it's kind of novel I
Yeah.
would love to know if there were other teams doing this. If
I
they
don't
are,
know of
I don't
any
know
other
who they are.
BioBuilder Club teams. So I don't know if there's like iGEM teams or anything that have done that. Okay. I will
Oh,
look
that's a
into
good
that. I'm
question.
curious.
That's a good question. I don't know.
I'm gonna look into that. I'm curious. Um, and another question that I have for the team is, this is a really cool option for BioBuilder Club teams to explore. Aside from the BioBits themselves that you had ordered, and your plasmid, what other supplies did you need? Since there's a lot fewer supplies and equipment needed compared to traditional methods.
We made our buffer.
Our buffer, yeah.
That's true. So our enzymes are thought to work better with buffer. So that was specific to the enzymes that we were studying. And then really the drying out for the SEM at the end. We just had an incubator and we used some dry right beads. But that was really it.
It's awesome that you were able to get that SEM data. I know that a lot of other BioBuilder Club teams don't necessarily have access to that. Can you kind of speak a little bit to that experience?
You said for the SEM?
Yeah.
So I think someone told us that they had a contact at UMass Lowell that worked with the SEM at the core research facilities. So we just reached out to them. And I think they were pretty happy to work with us. And this year we went back and we also used we worked with another lab manager for HPLC as well. So I think we just reached out. Their emails are readily found on their website and stuff. So we just went that way.
Yeah, and they were really happy to help and provide insights on softwares to use to analyze. So we've had our issues with ImageJ because we couldn't fully analyze, given that our scans had shadows and stuff that ImageJ did not really take into consideration when we were analyzing. So one of the lab managers helped us with finding another software that we would explore to hopefully find a solution.
And these guys don't know this, but we have another team that is also going to partner with them. They have a qPCR too.
Oh, cool.
Yeah, so we're definitely lucky to have them nearby. They're half an hour from here.
Yeah, another thing.
That's amazing. Yeah.
They also helped interpret some of the scans because sometimes there were these parts that were confusing. For one, there was a fiber and it kind of threw us off a little with the results. So yeah, the researcher there definitely.
That's great. Yeah, I think that, if teams are interested in collaborating with any core facilities at universities or even individual labs that you're aware of, the worst thing that they can say is no, right? So you should definitely send an email and ask, because it looks like this was really able to kind of extend your research and allow you to kind of start looking at the results. So that's awesome.
Yeah. And I'm so happy for these guys as students too, because I think that really is the nature of science. You know, we talk about scientists collaborating, but you know, looking at the thank yous at the end, how many different people really had to play into this to make it work.
Absolutely.
The true scientific experience.
Absolutely. Yeah. And, we get a lot of our mentors by reaching out to researchers. And I'd say, especially, everyone is always very friendly and willing to help students learn and have these experiences, which is awesome. Do you have any advice, team, for any other groups who are maybe interested in trying BioBits? I mean, I guess your advice could be go for it.
Yeah, definitely.
And everything takes longer than you think.
Yes, that is so
I would be
true.
like, oh, it's going to take us an hour to set this up, and we'd be here for 3 hours.
I heard that I caught that 9 hours monitoring it, where you guys actually in the lab monitoring it. Like what? I'm just curious what time frame you did that over.
So we actually use like the schedule send on iMessages. So like we just sent like 9 texts ahead of time. So everyone would just like be notified when to take a picture and put it on like a shared drive.
I love it. I love it. Collaboration is awesome.
Yeah.
Well, I'm excited to see the full presentation at the final assembly. And I really appreciate this. And thank you for sharing your information as well. If other teams are interested in BioBits and you didn't catch the emails, feel free to email me and I will connect you with the wonderful Andover team.
Great.
Thank you.
Thank you.
Thank you.
Awesome. Any last
sharing.
questions from? Oh, thank you, everybody. All right. So I'm going to stop the recording.